Hello, my name is Dr Nicola Robinson and I'm a Research Fellow at Canterbury Christ Church University. I'm working with a group doing research on epistemic insight. Epistemic insight is knowing about knowing and optimising learning and problem solving by drawing on interdisciplinary knowledge and skills, not just science. In terms of my career, I started out by achieving a degree in biotechnology, followed by a PhD in biochemistry. This led to me working as a research scientist at Pfizer Sandwich, where I worked with groups of scientists to identify new potential therapeutic drugs or potential new medicines. After working at Pfizer for around 10 years, I then became a science teacher, teaching chemistry as my specialty, and this led to me becoming a director of science at a couple of grammar schools in Kent. I really hope you like this activity I've put together, which you can do at home. I want you to be really thinking about working scientifically, so investigating, inquiring, critically observing. I also want you to appreciate that science as a discipline can be really powerful in answering questions and solving problems and making sense of our observations, but it does have limitations. However much we try to design experiments to answer questions, we know that we cannot fully control all the parameters involved. And this is obvious, I guess, when you do experiments at home and maybe at school, but actually it's important to be aware of this when you do laboratory experiments too. So good luck, but importantly, have fun too, thinking like a scientist and working scientifically. This activity is called Does Adding Fruit Affect Jelly Setting? It's an activity adapted from the Royal Society of Chemistry's Kitchen Chemistry, and you may wish to follow the link on the slide as Heston Blumenthal has a couple of related videos. I've provided a worksheet for this activity that you may wish to follow and use throughout, and I will signpost when you might wish to pause the slide pack and complete parts of the activity and the worksheet. Thinking like a scientist. So in order to answer the question, does adding fruit affect jelly setting, you'll need to think like a scientist. You'll need to think about what you will need to consider to answer the question reliably and whether there might be any limitations to your plans. I'll give you some time to think about your experimental design. You can pause here and use the worksheet to write down your ideas. I've provided a bubble map which you might find useful to organise your thoughts. Okay, so here are some questions that I thought would be good to think about when designing an experiment and answering a question. And as I go through each, compare them with yours. And I wonder if you had any other considerations that I hadn't thought about. So firstly, you have to consider what resources and what materials you're going to need to carry out the experiment. You're going to need whether you need to repeat anything. You need to consider what you need to control in order for this to be a fair test and for you to generate valid data. You should think about what you will be observing, what observations or what measurements you're going to make, what type of investigation are you going to perform. Are you looking for patterns or changes in something over time, or are you just looking to observe something happening or not happening? How are you going to collect your data so that you can then share it and present it to others? And also, how will you actually know, once you've done your experiment, whether you've been able to answer the question successfully? You'll see a table, and I'm going to give you another pause point so that you can think about the resources and the materials that you're going to need to answer the question about jelly setting. You can then compare your ideas with mine in a few minutes. So this table is on the worksheet and it's called Task 2. So pause the slide pack now so that you can fill in the table. If we now look at the filled in table that I prepared earlier, you've had time to think about the materials and methods, and so now we can compare our ideas. Remember, we're looking at whether jelly setting is affected by fruit. So it's obvious, I suppose, isn't it, that we need to have jelly, we need to have fruit, and we need to have some kind of container to put the fruit and the jellies in. One question I had was, why would it be advisable to use more than one type of fruit? What if different fruits had different effects on the setting of jelly? If you only used one fruit, it would just not be possible to assess this, would it? Then let's look at the method. I thought of 2 different methods, you might have thought of more. So method A is where you would make up the jelly and the fruit together before allowing them to set. Method B is where you make up the jelly as per the packet's instructions and then lay fruit on top. So this second method looks at whether the fruit can affect the setting of the jelly after it has originally been set, whereas method A is looking at whether the fruit affects the original setting process. So working scientifically, when we are trying to answer questions scientifically, we need to consider whether we're performing a fair test. Are we able to change just one parameter whilst keeping all others the same? Let's consider method 1 from now on, and if you want to investigate method 2 independently, then that would be amazing. So method 1, we need to substitute some of the water that would be used to make up the jelly in order to incorporate some fruit. So that means we're going to have to make sure that the volume of the water removed from the jelly recipe is the same for all the different fruits that we test. And the other thing that we need to keep the same is the amount of fruit for each test. And what do we mean by amount? Do we mean mass? Do we mean volume? What do we mean? We should also keep the setting time and the setting temperature the same and use the same type of jelly throughout. But how difficult is it going to be to do this at home? I'm going to refer to the parameters that we need to keep controlled as control variables. Now, the ones that are probably going to be quite easy to control are things like the setting time, the temperature that you set the jelly at, and the actual jelly itself. So I think it's going to be quite easy to control those variables. Using the same container, well, instinctively you might think, well, that's going to be easy, but at school, yes, it probably would be easy. At home, will you have enough of the same container? And then keeping the same amount of fruit per test will be tricky. How are we going to control the size, the volume, the mass? Especially bearing in mind that different fruits have very different consistencies. Electronic or weighing scales in homes are usually designed for larger masses than would be used here. So we don't really have the equipment with the appropriate resolution to make these accurate measurements. So can you see that whatever we observe in our experiments is going to be limited by the equipment and the resources we have available? We're going to pause again now so you can set up your experiment, but you may want to watch my video first so you can see how I've set up mine. Okay, so in order to start this experiment, because we're working to see whether the jelly sets with different fruits in it, what you have to do is prepare a little bit in advance because obviously it takes a bit of time to set. So I've got some standard jelly, strawberry, my favourite. And basically what I've done is I've taken off each of these cubes, I've pulled them apart like this and put them into a jug. And according to the instructions, it basically says to make it up to half a pint with hot water. Do be careful. Make sure it dissolves, and then you're meant to make it up to a pint before you actually pour it into a mould and get it to set. Now, because we're using some fruit, we're going to have to substitute some of that extra water. So what I've done is make it up to 3/4 of a pint, pour that out, and then add bits of fruit to it. So if you have a look over here, this is what I've prepared in advance. So what I've got as our control is I've got some glasses here which has just got jelly with the full amount of water. So it's basically got made up to a pint of jelly and water. And then all the others have had less water in, but they've been made up with either fruit or fruit juice. And so if I take you through what we've got, so as I said, the first 3 on the right there are the controls. Then we've got jelly that's been made up with pineapple juice, and that's fresh pineapple juice. The next one we've got is made up with fresh pineapple. You can see it floating on the top, yeah. The next one I've got is tinned pineapple. Now that's a bit interesting, and we'll have a chat about that later. I've then got some apple. I've got blueberry, kiwi, strawberry,
papaya and orange. So I think that's quite a good selection of fruit. And don't worry, we'll eat it all at the end, so nothing will go to waste. So what I wanted particularly to show you, to prove to you that I'm not cheating here, is that they are all wobbly at the moment because they're all in the liquid form before I let it set for a couple of hours in the fridge. So if I just pick up a few randomly, you can see they're all very, very wobbly. And so hopefully when we come back to this in a couple of hours, we'll see a bit of a difference. Okay, so hopefully you've set up your experiment. Since jelly does need some time to set, there's going to be a gap between you setting up the experiment and making your final observations. But what we're going to do now is look at the results of my experiment, and it would be great if you can compare your results with mine. So I've included another video here with my results. Okay, so here we are back after the jelly has been allowed to set for a couple of hours, and I have to confess that I've left it in the fridge overnight, in fact, so it's had considerable time, so we should see the maximum effects here. What I didn't say yesterday was the fact that I've done each of these little tests in threes. So for each of the combinations of the jelly and the fruit, there are 3 of each, and I really want you to think about why that's important. Why is it important not to do just 1 or not even just 2, but why is it important to do 3 of each test? Okay, so now let's go through all the results and let's see if there's anything surprising. So let's look at our control experiment first. So this is the one where we've just got the jelly with a normal recipe amount of water, and you can see that it's completely solid. Yes, and if I attack it with my spoon, you can see that, yeah, it is solid. So let's have a look at the other combinations. So the next combination was the fresh pineapple juice from a carton. And this one has actually surprised me, but we can talk about this a bit later. But if I put my spoon into this again, and I'll turn it upside down, you can see that actually it has set. Now, let's have a look at the next one. The next one is where we've got the fresh pineapple, and you can see that I'm wobbling it around here, and in fact, it hasn't set at all. So if I show you this, I can pour it, there we go, straight into this bowl here. It hasn't set at all, and that's the one with the fresh pineapple. We also used tinned pineapple, so from a can, and this time you can see again that it's actually set. So I can turn it upside down and it is set. So the fresh pineapple has not set, the tinned pineapple has set. If we look at the next combination, we've got apple, and you can see here that that hasn't affected the setting at all. And maybe you want to, as an added observation, notice that the fruit is relatively low density. It's sitting on the top of there. It hasn't sunk to the bottom. We look at blueberry, again, hasn't affected the setting at all. Kiwi, oh, now then, have a look at this one. So kiwi as well has not allowed the jelly to set. So there we go, I can pour that just to demonstrate that hasn't set at all. If we look at strawberries, so strawberries, they've set. We've then got papaya, and you can see again with papaya that hasn't set either, and I can pour that straight away into my bowl also. And then lastly, we've got orange. So orange, again, is one of the fruits that has allowed the jelly to set. So we need to sort of unpick all these results and try to understand why, or come up with some hypothesis, explanation why some fruits are allowing the jelly to set and some fruits are not. You can also see from this slide that I've got a table and that's the way I collected my data, where I put the different type of fruit and whether or not the jelly set. Isn't it interesting that the results here suggest that different fruits have different effects on the jelly setting? If you look, fresh pineapple, kiwi and papaya didn't allow the jelly to set. I wonder why that is. I also wonder why the tinned pineapple and the pineapple juice didn't affect the setting, whereas the fresh pineapple did. Now you are going to think about the observations made and what you can conclude from those observations. So that's task 3 in your worksheet. Can you actually answer the original question, does fruit affect jelly setting? Is there anything that concerns you about how reliable the data is? Could we have had a better experimental design perhaps? So again, I'm going to pause, or you can pause here, so that you can complete tasks 3 and 4. So now, if we look at the experiment conclusions. In terms of conclusions, we can definitely state that the fresh pineapple must contain something that affects the jelly setting, and that the strawberry and the tinned pineapple didn't. And obviously we can say similar things for the fruit according to what fruit you tested. Thinking about the tinned pineapple, this has been prepared by heat sterilisation as part of the canning process. So maybe the something in the fresh pineapple gets destroyed by heat. Can you think of anything natural in fruit that could be destroyed via heat treatment? You need to think about biology and chemistry here perhaps. So fruit contains enzymes that will have been destroyed or denatured by heat. Maybe an enzyme in fresh pineapple affects the jelly setting. Come up with some explanations. Let's think about jelly first. How does it actually set? Well, jelly contains gelatin, which is a protein, and it's a processed form of collagen. Collagen is the most common protein found in humans and other mammals. When the gelatin is dissolved in hot water, the bonds holding the collagen protein together are broken. Then, as you allow the gelatin to cool, new bonds form, trapping the water between them, and thereby they form this semi-solid, which we know as jelly. So if we think now, trying to put everything together, we know that jelly contains a protein called gelatin, and that allows jelly to set. And fresh pineapple contains some enzyme, and if you do some research here, you'll find out that fresh pineapple contains proteolytic enzymes, so they break down proteins. In the case of pineapple, the protein is called bromelain, and that breaks down gelatin, and therefore the jelly can no longer set. And what I found when I used kiwi and papaya was that the jelly again was affected like the fresh pineapple, and that was because they also contain proteolytic enzymes, and they also break down the gelatin, disabling the gelatin's ability to act as a setting agent. So it's interesting, isn't it, why pineapple is different according to whether it is fresh or tinned. So for the tinned pineapple, we did previously discuss, didn't we, that in the canning process, the proteolytic enzyme would be denatured because of the heat treatment. What about the fresh pineapple juice though? Why didn't that prevent the jelly setting? Well, some reasons I've come up with here are that maybe the fresh pineapple juice in cartons has to undergo some kind of sterilisation, so it's basically a similar story to the tinned pineapple. Another reason I've got is maybe the enzyme concentration wasn't sufficient in the fresh juice. Maybe I didn't substitute enough of the juice in the jelly making process. Maybe the juice in the carton contained a chemical or a preservative that affected the enzyme. So in terms of what we could do next, can you think of anything else you'd like to try in order to explore the investigation further? Doing some research on this, I came across something interesting in that I read that if you add chili, that will prevent the effect of the fresh pineapple on setting, and that chili contains a chemical that destroys the enzyme that prevents the jelly from setting. So that could be another interesting factor to investigate. Other things you could look into are alternatives to gelatin. So gelatin is derived from animals. There are some algae derived gel setting agents, and you might have come across these in regard to sort of vegan products. I wonder whether fresh pineapple would affect the setting of those products. So it's reflection time now. Let's reflect on what you've done so far. So you've looked into whether different fruits affect the setting of jelly. You've practiced thinking like a scientist, designing experiments, making observations, drawing conclusions, evaluating your experiment and thinking of explanations for your results. You've also experienced the power of science in being able to answer a question, but you've also experienced the limitations of science and how difficult it is to control all the variables in an experiment, especially at home. So let's think about linking all this as well to material science. So we've talked about gelatin in the jelly, and this has some interesting properties. It's a material known as a biopolymer or a hydrocolloid. Hydrocolloids are a heterogeneous group of long-chain polymers, and you would have come across polymers before if you were learning about plastics. But natural polymers such as polysaccharides and proteins also exist. And these hydrocolloids are characterised by their property of forming viscous dispersions, so thick dispersions, and gels when dispersed in water. And this is because they have a large number of what's called hydroxyl or OH groups, and you might have come across those when you have learnt about anything to do with alcohols, which have OH groups. And these markedly increase their affinity for binding water molecules, so that makes them hydrophilic. So hydrophilic means liking of water. Furthermore, they produce a dispersion, which is an intermediate between a true solution and a suspension, and so that's why it's called a colloid. So these are interesting materials, and they're used in many applications. You might have come across them in terms of biodegradable packaging. They're often used in the food industry to affect textures. They're used in cosmetics, pharmaceuticals, even in photography. And as I mentioned before, there are some non-animal derived versions available. Thinking about other materials, so we talked about enzymes as well, didn't we? We talked about the enzymes in pineapple, such as bromelain. These enzymes break down proteins, hence they're called proteolytic, and their properties have been exploited in food industries. If you look at the diagram, you can see that they're used in cosmetics and textile industries too. For example, as teeth stain removers and to treat silk and wool. Apparently they improve the quality of dyeing those materials. Interestingly, perhaps, due to the increased demand for these enzymes across many of the different industries, scientists have now been working on improving the production or the mass production of these enzymes, and have even used recombinant technology to make the enzymes synthetically. So I really hope you've enjoyed doing this activity and thinking like a scientist. I hope it stimulated you to take the ideas further by designing other experiments to perform. I also hope it's made you more aware that yes, science as a discipline can be really powerful in answering questions and making sense of our observations. But science can have its limitations too, both at home and in science laboratories. Being aware that science has its limitations in answering questions is very important, as it makes you understand the need to provide a context to your results and your conclusions, so that when you share information, others are aware of the limitations too. I hope you found it interesting too, learning about materials such as enzymes and hydrocolloids and gels. And lastly, I'd like to thank you for performing this activity. Maybe you can get your friends to do it too, so you can discuss ideas concerning the science here and also how to optimise the experiment further. Whatever you do, have fun.